Oddo. He works for physiology over at UTHSCSA Graduate School of Biomedical Sciences. He's been an assistant professor since July of 2008, and it was September that same year that he gave his first grand rounds talks here with us for mouse models of AD. Dr. Oddo earned his BS in biological sciences from the oldest university in Sicily. Once in the U.S., he garnered a PhD in neurobiology and behavior at UC Irvine. The major objective of his lab work is to elucidate the molecular basis of cognitive impairment in Alzheimer's disease and other related neurodegenerative disorders. To that end, some of his best research has accumulated into the results that you'll see today in his talk. So please give him a warm welcome, Dr. Oddo.
Okay, so thank you so much. So the goal of my lab, like you mentioned, is to understand the molecular basis of cognitive decline in Alzheimer's disease. And for that, we use animal models. And especially right now, we're focusing on mTOR signaling, and I'm going to tell you why in just a little bit. So, just a brief overview of the talk. I'm going to give you a brief background on Alzheimer's disease, just so everybody's on the same page. And then I'm going to tell you why we study mTOR and what is its relationship to Alzheimer's disease. And then I'll go into the meat basically of the talk and show the data we have generated over the last few years here in Salvatore. So Alzheimer's disease is the most common neurodegenerative disorder in the world. It is currently the 6th leading causes of death in the United States. And about 5.5 million people are affected. And this number is basically, it is estimated by the Alzheimer's Association about every 70 seconds is a new diagnosis of Alzheimer's disease. And so these numbers will get only worse because aging is the major risk factor of Alzheimer's disease. And as the aging population is booming, and as the aging population is booming, these numbers are going to get worse. And it's estimated that by mid-century, this will be more than double. The number of people affected by Alzheimer's disease will be more than double. So if you think that sequestration is a big financial issue, think again. Pretty much. Okay, so as I mentioned before, Alzheimer's disease is the 6th leading cause of death in Alzheimer's disease. These are the top 5, and these graphs reports the number of deaths between 2000 and 2006. And so as you can appreciate, there has been basically an improvement, if you will. Right? Fewer deaths from HIV, heart disease, breast cancer, prostate cancer, stroke. However, when you compare that to Alzheimer's disease, again, in 6 years, there's been a 46% increase in number of deaths. And this is only going to get worse because the number of people with Alzheimer's disease is greatly increasing because the more and more people are over the age of 65 or 70.
Well, is that number because we got higher or better life expectancy?
Yeah, that's one of the reasons. Because basically aging is the major risk factor of Alzheimer's disease, and as, for example, the risk of developing Alzheimer's disease in people that are 85 years of older is almost 50%. So when this fraction of population increases in any country, you see the more people with Alzheimer's disease.
But it's say we only, I think the life expectancy here only increased from 76 to 77 or 78 something, but there you got 46%.
Right, because there's nothing, there are no cures for Alzheimer's disease, there's nothing that can slow the progression of the disease. And here you get an improvement because of the care that physicians and science has, or the impact of science and physicians on this disease. And right now there's nothing for Alzheimer's disease. So from a pathological point of view, in this cartoon, you have half a brain from a person with Alzheimer's disease, half a brain from a healthy control. And you can immediately see that there is atrophy of the brain, all the ventricles are bigger, and this is due to profound cell loss throughout the brain. Can I turn off the light? Is it too dark? No. So at the macroscopic level, the brain of people with Alzheimer's disease is characterized by two major lesions. These amyloid beta or A beta. These are extracellular plaques and intraneuronal inclusions that are called neurofibrillary tangles. And these are formed of the microtubule binding protein tau. And these are formed by this peptide called amyloid beta. And this peptide, amyloid beta, is derived from a larger precursor called amyloid precursor protein, or APP. In this cartoon, this APP is a transmembrane protein, and the amyloid peptide is this red box. So normally, this is physiologically expressed in every cell, and normally it undergoes through what is called an alpha secretase process, or a non-amyloidogenic process, where an enzyme called alpha secretase will cleave this precursor in the middle of the beta sequence, therefore precluding a beta formation. And so you can see here in this cartoon icon, A beta cannot be formed. However, in pathological cases, the first enzyme that cleaves the precursor is called beta. It's called beta secretase, which cleaves at the amino terminal of the A beta sequence. And now this beta stop, or C99, which is 99 amino acid, is now a substrate for another complex, another enzyme, which will liberate A beta. And this is believed to be the toxic species. Accumulation of this is a primary event in the pathogenesis of Alzheimer's disease. Basically, only a few cases of Alzheimer's disease, far less than 5%, are linked to genetic mutations. And all the mutations that are found so far, all in the proteins, are related to this pathway. For example, there are mutations in the amyloid precursor protein that are lead to Alzheimer's disease, and they are 100% penetrant. There are mutations in this gamma secretase, which is presenilin 1 or presenilin 2, and also those mutations are 100% penetrant. And all of these mutations lead to an increased production, an increased level of A beta 42. The other protein, as I mentioned before, involved in this at the neuropathological level is the accumulation of this tau. This is a microtubule binding protein. It's normally involved in stabilizing the microfibrils. And in Alzheimer's disease, it is hyperphosphorylated. And when it is hyperphosphorylated, it comes off from the microtubule and will accumulate to form these neurofibrillary tangles. Genetically, mutation in tau lead to a different form of dementia. It's called frontotemporal dementia with Parkinsonism. However, the neurofibrillary tangles, these people also characterized by the accumulation of neurofibrillary tangles that are similar to those that are found in people with Alzheimer's disease. So in my lab, we use animal models to study the basic mechanisms of Alzheimer's disease. And the mouse model we're going to use today, I'm going to talk through the rest, is called 3XTGAD. We developed this in Irvine, California, about 10 years ago. These mice are characterized by the accumulation of amyloid beta, and these are different techniques used to show A beta accumulation by immunohistochemistry. Here you can see throughout the hippocampus all these brown deposits. Or you can use histological stainings, again, to see these A beta plaques. These mice are unique because at the same time, in similar brain regions, they also accumulate neurofibrillary tangles. Again, these are formed by tau accumulation and phosphorylation. And again, here, just to summarize, we can see these neurofibrillary tangles using different histological methods or by EM. Again, these mice have been around for more than 10 years and have been truly characterized. These are other neuropathological features that are developed by these mice. And what's most important for the rest of the talk, these mice develop age-dependent cognitive decline. And so I'll show you some of those in a little bit. So that brings me to mTOR signaling. Again, our goal is to study signaling pathway that may account for alterations in learning and memory in these mice and in Alzheimer's disease. mTOR signaling is part of a complex, in this case mTOR complex 1, and basically regulates both protein translation, a positive regulator of protein translation, and is a negative regulator of autophagy, or protein turnover. Therefore, by regulating both protein synthesis, protein translation, and protein turnover, it regulates the amount of protein that are in the brain, in any cells. And so it's important, and so it may play a role in any proteinopathies, in any disease characterized by protein accumulation. There is more. Randy Strong here at the VA and Barshop with his colleagues through the NIH Intervention Center a few years back showed that mice that were fed rapamycin, which is an mTOR inhibitor, will live significantly longer than mice on a control diet. Okay? So there is this improvement in lifespan. A few months after that Nature paper, another group came up with a genetic approach to actually, again, reduce a little bit mTOR signaling, and they saw, again, the same similar results, an increase in lifespan, and actually, in this case, also health span, and improvement in behavior function of these mice. These are P70X6 knockout mice. This is a protein directly downstream of mTOR. And actually, its phosphorylation level are normally used as an indirect measure of mTOR activity. So before we got into this mTOR signaling Alzheimer's disease, there were already data that show that mTOR was upregulated in brains of post-mortem brain from AD tissue. In this sample from this paper in 2003, you can see they have a Western blot where they measure the total and phosphorylated level of this P70X6 kinase between AD patients and control. And you can see there is more, the phosphorylated level of this P70X6 kinase are significantly increased in AD brains. And again, this is an indirect measurement of mTOR activity, because mTOR directly phosphorylates this protein at this epitope. And so these data were confirmed by several groups. However, what was not clear was basically the question we wanted to ask at that point is, what is the relationship between A-beta and tau with mTOR? Does it play a role in the A-beta-induced cognitive decline? Does it play a role in the tau-induced cognitive decline? And those studies are hard to do in people because you can't do much manipulation. So what we decided to do, we used our animal model, and first thing we did, we measured mTOR signaling. And here, basically, we use a commercially available kit to directly measure mTOR activity in 3 brain regions of these transgenic mice that we use, and at two different ages. Here in white, you have the wild-type mice, so non-transgenic, that we use as a baseline, and then in black, you see the mTOR activity in the transgenic mice. So both as in 2 out of 3 brain regions, we see an increase in mTOR activity both at 6 and 12 months of age. In the cerebellum, we saw the mTOR activity was similar between the 2 groups. And this basically correlates with the amount of A-beta, with the distribution of A-beta pathology in the brain of these mice. Indeed, the hippocampus and cortex are 2 brain regions highly affected by A-beta pathology. However, the cerebellum doesn't have any A-beta pathology. So it serves as a nice internal control. To go behind the correlation, what we did, we removed the beta from the brain to see if there was sufficient to restore mTOR activity. To do that, we use an old technique. A few years back, we showed that a single intrahippocampal injection of anti-A-beta antibody is sufficient to clear A-beta from the brain. And so you can see here, again, in this example, we can inject A-beta antibody. Here, a single injection, this is the cannula tract, a stereotactic injection. And you have clearance of A-beta deposits in the brain, just in the brain region receiving the antibody, not in the control artery. So you can appreciate the difference here. So we've done the same thing here in San Antonio. When we moved here, again, we can measure A-beta levels. In the ipsilateral side, the side receiving the antibody, we have more than half, there's a significant decrease in A-beta levels.
How long, I'm sorry, how long does this take? How long?
This is, in this case, it's 24 hours after single injections. We were not the first one to show this. We used it as a tool back then to see what would happen to the tau pathology after removing the A-beta. Here, we're using it to see what happened to mTOR after we remove A-beta. Other people have done extensive time course of this A-beta immunization. Now there are ongoing clinical trials. There are failed clinical trials, there are ongoing clinical trials for A-beta immunization. But the point here, we're using it as a tool just simply to remove A-beta from the brain and look at mTOR signaling. When we do that by Western blot first. And so this is again, we measure the P70X6 kinase levels, total phosphorylated. And you can appreciate we have about a 50% decrease in the level of P76 kinase in the side receiving the antibody. So lowering A beta will basically decreases mTOR signaling. When we directly measure mTOR activity and compare to wild-type mice, again, the control arm uninjected, these mice have an increase in mTOR activity. When we put an A beta antibody, we lower the A beta levels. Now the mTOR, activity is similar to wild-type mice. Okay. To go behind that, we actually wanted to test whether A beta was sufficient to cause mTOR hyperactivity. We use now wild-type mice. These are 2 months old wild-type mice. And what we did a stereotaxic injection of a conditioned media from this CHO cell line that's highly enriched in A beta. This cell line is stably transfected with ATP, and it's known in the field to secrete A beta in the media. So we can concentrate the media, injected stereotaxically in the hippocampus of these wild-type mice, and measure A beta, and measure mTOR signaling. As a control, we have conditioned media from a parental line, it's untransfected CHO cells, or conditioned media from the 7PA2 cell line, again, it produces A beta, where A beta is immunodepleted before the injection. So this approach has been used in the past, a few years back now, and show that a single injection of this media reaching A beta is sufficient to cause memory dysfunction in mice after a single injection. Okay? So we did it again in wild-type mice. 72 hours later, we took the hippocampus out and measured mTOR signaling the same way. Now this is throughout basically the talk. I measured mTOR signaling by measuring P76 kinase, both total and phospholipid. So if you look at the quantification of the blood here, when we inject the A beta containing media in wild-type mice, here we have an increase in phospholevels in the phospholevel of P76 kinase compared to the hippocampi where we injected the CHO media, the control media, or compared with the hippocampi where the same media was immunodepleted before the injection. So we can remove A beta before injecting it. And this again correlates, this mTOR signal correlates with when we directly measure mTOR activity, we see the same thing. And injecting A beta into wild type, A beta containing media increases mTOR activity, but removing A beta from the media before the injection actually doesn't have any effect. So these data so far, that I showed you so far, is that A beta is necessary and sufficient to cause mTOR hyperactivity. What we ask next is, what is the molecular mechanism underlying this A beta-induced mTOR hyperactivity? And before I go into that, I want to bring back this cartoon and show you and point your attention to this protein called Plus40. Plus40 again is part of this mTOR complex, and when it binds when it's bound to this complex, it has a negative effect. So it reduces the mTOR activity. When Plus40 is phosphorylated, it comes off from the complex, and now mTOR is more active. And usually Plus40 is senses. It's normally regulated through insulin signaling, which also has been linked to Alzheimer's disease. And when you need, after high insulin signaling, will basically phosphorylate this comes off and you have a more mTOR activity. So what we did, we measured Plus40 total levels and phospholevels, again in the transgenic mice, and we see an increase, you can see here in the quantification, of phospholevels of Plus40 in these mice, indicating that this might be, that the A beta-induced mTOR activity might actually go through this. Remember, when this is phosphorylated, it comes off from the complex, and mTOR is more active. How did we also have data from human brains, and we measure, these are very preliminary, and we measure Plus40 levels, phospho-Plus40 levels, in 4 groups. These are, basically, let me tell you what these groups are. These are people, all age matched, controlled, cognitively intact. These control two are people that are cognitively intact but have high beta load in the brain post-mortem. These are early AD cases, and these are people with AD with more advanced AD. And so it's a nice trend. There is nothing statistically significant yet, because we have very few ends. But again, it's a nice trend as we basically, the disease is more severe from left to right, and you can see this nice trend. So we are getting more tissue. But to see, again, this is just a correlation so far. To go behind the correlation, what we did, we basically have redone the experiments where we inject A-beta into the brain of wild-type mice, and then at this time, at the same time with the A-beta containing media, we inject an AKT inhibitor or a PIM1 inhibitor. These are the two only known kinases that phosphorylate plus 40. So we inject in A-beta, and then at the same time try to prevent plus 40 phosphorylation. And they work quite well, otherwise I won't be here to tell you. And so when we inject 7PA2 media, we have again confirmed we have an increase in an increase in plus 40 phosphorylation. And then plus 40 phosphorylation, however, does not change when we inject A-beta media in the presence of these two inhibitors. And this basically it nicely reflects the mTOR activity. Again, when we have high plus 40 phosphorylation, you have more mTOR activity. When you have low plus 40 phosphorylation in the presence of A-beta, you have less mTOR activity. Again, suggesting that some of the A-beta induced mTOR hyperactivity might go through this plus 40 phosphorylation. And we are trying now to, we're doing some pharmacological experiments that I have no time to show you. They're very preliminary, where we may have identified actually a small molecule that can reduce plus 40 phosphorylation. And we will test those in mice. So what I showed you so far is there is that the A-beta is necessary and sufficient to cause mTOR hyperactivity, at least in mice. And it's known from, there is a large body of literature showing that A-beta can induce cognitive deficit. So the next question we wanted to ask is whether the A-beta induced cognitive deficit, whether does it go through mTOR? Or it's basically the A-beta induced mTOR hyperactivity that's nothing to do with cognitive decline.
So, before you go on, the A-beta, I think, say when you inject into the mice, you inject into the inside of the cell or the...
I injected in the, so we go in with a needle through the brain.
Yeah, but say if it's outside of a cell and the mTOR is inside,
Yes.
so
we have, we're working on, so one aim of my grant is to follow up on this plus 40 phosphorylation. And we have some idea what A-beta can bind to, for example, beta-2 adrenergic receptors. And this can, through ERK signaling, can alter mTOR activity. So it's from the outside, it binds to specific receptors. We try to identify and dissect that pathway. But mostly I would just speculate. We don't have strong data.
Also, do you think that these effects are specific to neurons, or do you also think that there could be astrocytes?
We have to look at it. But so what we could, if we get money, because we have mTOR. Now we have. I'm getting ahead of myself. Hold on. Yes.
Do you have data from sham injections? Because mTOR should be activated by trauma.
So these are injections. They're not sham, but we do inject media without A-beta, and so that's our control. So they should control for the trauma. Okay. So the next part is we wanted to see again if the A-beta induced mTOR hyperactivity contributes to cognitive decline or has nothing to do with that. Okay. To do that, we treated these transgenic mice with rapamycin. Again, it's an mTOR inhibitor. Rapamycin was given to the mice via with the food. And this is the same diet that Randy Strong and colleagues have used in their lifespan studies. So we've done these experiments basically two different cohorts. In the first cohort, first group of mice, we have transgenic and non-transgenic, either on rapamycin or on a controlled diet, where rapamycin was given at 6 months of age for 10 weeks. Okay? So this is the onset of cognitive decline in these mice. That's why we chose 6 weeks. In another group, in another independent study, what we've done is we gave rapamycin at 2 months of age for 16 months. And so this is to determine whether it can prevent the onset of pathology and the cognitive decline that these mice develop throughout their lifespan. In the 2 months, it's basically they have no pathology, they basically they are indistinguishable from wild-type mice. We have another group of mice where they were kept on the control diet for 15 months, and they were given rapamycin at 15 months of age. This is more like of a treatment group, because at 15 months of age, these mice have cognitive decline, they have widespread pathology in their brain. And then we have another group of, they stay on the control diet throughout the lifespan. And for these 3 groups, we have wild type and transgenic. So we have 6 groups in total. Okay. So there is not enough time to talk about this. So we published this now 2.5 years ago or so. And so I can answer a question at the end, or you can just go look at the paper. So I'm going to show you the results of this experiment now. Okay. So first thing is we actually have data with Randy Strong actually has data. The rapamycin does cross the blood-brain barrier. These are another transgenic line treated the same way, where they measure by HPLC, they measure the level of rapamycin in the brain and the blood. But my point is, it gets to the brain, crosses the blood-brain barrier, and it can be detected. And that's basically unexpected, because again, if you dig in the literature, you find data or papers where they show that this rapamycin does cross the blood-brain barrier. Okay, so the first thing, it does cross the blood-brain barrier. What we wanted to do, we wanted to see if it actually reaches the target, the intended target, and has any biological activity. And as now you're familiar now with this, we measure P76 kinase levels, total phosphorylated. And now we have 3 groups. These are just the transgenic mice on the control diet, treated for 3 months, 15 to 18, and treated for 16 months, 12 to 18. Okay, so this is the same pattern you see throughout the talk. And so, as you can see, rapamycin is given either prophylactically or as a treatment, but it basically reduces mTOR activity in the brain of transgenic mice. Okay? So, after that, we tested the mice in a behavioral task to see if there was any effect on learning. To do that, what we used in this case is called Morse-Water maze. I briefly go to the task. This is basically a tank about 1 meter in diameter, where we put a mouse in the tank. They don't like to swim, so they swim around randomly until they find a platform. This platform is just an escape. It's under the water surface. The water is opaque. We put non-toxic paint, the same paint that my kids play with. And so they can't see the platform, but they can bump into it because it's literally underneath the surface. So if they swim randomly on top of it, they bump into it and climb. So they don't have to swim. They get out of the water pretty much. So that's an escape. So if we do that long enough, eventually mice or rats in this cartoon will swim directly to the platform. And the way they learn it is by navigating, because there are cues, extra nasal cues around this platform. And so they basically, using these reference points, they will learn what the platform is without actually seeing it. And this is an hippocampal dependent task. It's like you finding your car in a parking lot or in a parking structure. Basically, it's the same principle. Okay, so here I'm showing you actually now real data. Just 2 small videos, I think I have to go here. And these are transgenic mice on rapamycin, transgenic mice on the control food. They've been trained for the same amount of time. And here, just let's look at this. They've been treated with rapamycin, they swim around, they look, they find the platform. When they find the platform, the person doing the experiment takes it out as a reward, and they go in a nice warm cage. The video here quit, but basically, usually if a transgenic, the video keeps quitting. I'll show you the real data. And so basically here, in this is in the corner, there's a wall here, and there are cues. Basically, the mouse can see, the person doing the experiment sits in the same place because that's also a cue. And again, basically we'll navigate this maze and find the platform they're actually seeing. And this is how we plot the real data. It's escape latency is the time to find the platform, and here is how many days of training. So this is the average of 4 training trials for each day. Okay, so we have in these shades of blue, we have the transgenic mice, red, orange, yellow, we have the wild type mice. So these are the transgenic mice from the control diet, which in this case, basically they have no much improvement over 5 days of training. They go from 50 something at 48 to 30. When we compare them with the two group of transgenic mice on rapamycin, we see that when we give rapamycin as a treatment, if you start rapamycin at 15 months of age, there is no improvement. This is the line, basically. There's no improvement in learning this spatial task. However, when we give rapamycin, we start the treatment 2 months of age before any pathology. Now these mice eventually will perform better, so we'll find the platform faster compared to wild type. Compared to transgenic on the control type. Similar results are found with wild type mice. So now I have all these groups together, it's hard to follow, but the non-transgenic in red are basically the baseline, the non-transgenic on the control diet. And this actually, rapamycin also improves performance in wild type mice. So it could be used as a, based on that data in mice, could be used as a therapeutic approach for age-dependent cognitive decline, not necessarily related to Alzheimer's disease. So after 5 days of training, we basically put the mice back 24 hours after, we put the mice back for one last time for 60 seconds. This time we remove the platform. Okay? So just to see if they remember what the platform used to be. And so a mouse will remember, if I can draw it here. This is what the platform used to be, for example, right? And so if a mouse will swim there, it doesn't bump into it, so it tends to look around where the platform used to be, something like that, okay? So what we do, you can split the platform in 4 quadrants, and you can measure during 60 seconds how much time they spend here compared to the other quadrants, okay? So if it's random, you will see 25 seconds each on average. If the mice know where the platform used to be, they will spend most of their time here. We also can measure number of time over 60 seconds that they swim over the platform location. Okay? And so all this is unbiased because there is a software that actually gives us an Excel sheet. So this is being taped and there is a tracking system that just gives us an Excel sheet. So in this case, I'm just showing you the number of platform location crosses. So how many times it goes where the platform used to be. On this side, you see the wild type mice. And so you can see a regular 18 months old mouse doing 60 seconds, about on average 3.5 times crosses the platform location, about 3.5 times. However, when we give rapamycin to these mice throughout their life, basically we have a significant increase. We see a significant increase in number of platform location crosses, indicating they remember better. The implications they remember better.
Why do you think the non-transgenics that you give rapa is still even better than the transgenic ones?
So it would be the red this versus this?
Oh, no, the green one. Non-transgenic rapa 2 to 18 in comparison to light blue 2 to 18. So
These guys?
yeah, that one, the other one, that one even better than the transgenic. In
Right.
other words, is there any other genetic
I'll show you.
things?
No,
Oh.
hold on, give me
Sure,
3
that's fine.
minutes. I think it's coming. The answer
Okay.
to your question. Okay, so what we believe, basically there's an improvement in both transgenic and wild type only in mice that receive rapamycin at 2 months of age. What we believe that the mechanism underlying this improvement are different from the mechanism underlying this improvement. And again, there's not enough time to show you both, so for the wild type mice, I just refer to you to this paper we published about a year ago in HHS-HRSA, or we can talk after. So I'll spend the rest on this group of transgenic mice and I'll answer your question. And I guess the answer is here. So first let me go through this. This is now after the behavior. Mice were sacrificed, we take the brain and we look at the brain for pathology. By pathology, I mean A beta deposits in this case, where again, this you read it by columns. You have transgenic mice on the control diet, transgenic mice on rapamycin for 3 months, transgenic mice or rapamycin for 16 months. So you can appreciate, without me telling you anything, you can appreciate there are fewer deposits of A-beta deposits, basically, in the mice that were treated with rapamycin prophylactically compared to the other two groups. And here, rapamycin did not clear A-beta, So to answer now your question, is these mice still have A-beta deposits. So these, even if there is an improvement compared to the transgenic, so if I go here, even if these mice have an improvement compared to these, you know, they still have A-beta deposits, still have deficits, and so it's very, yeah, they don't go back to baseline, basically. They don't perform as well as wild type. Most likely because of these residual deposits of A-beta. So we also measure A-beta biochemically, and we can see similar results. I can quickly skip this for time's sake. Next, we look at the tau pathology. And now these are, again, hippocampal sections, CA1 pyramidal neurons. And you see the same pattern in the mice, two different markers of tau, but it's the same pattern with A-beta. In the mice, they're given rapamycin after the onset of the pathology. Rapamycin has no effect, okay, compared with the control. However, when we give rapamycin prophylactically, now you have fewer neurons that are positive of these tau markers. Okay? So now, if you remember, mTOR activity was decreased in both groups compared to the control. So there is this dissociation between mTOR activity and effect on pathology. Okay? And we'll go into that in a little bit. So with as I mentioned earlier in the talk, mTOR is a negative regulator of autophagy, which is involved in protein turnover in A-beta and tau turnover. And so this cartoon shows what A-beta does, what mTOR does basically, negatively regulates one of several of these autophagy-related proteins. They are necessary for the autophagosome formation, for autophagy induction. When this autophagosome is formed, basically it will form around proteins or organelles that have to be removed. It will fuse with the lysosome for protein removal, for digestion, basically, and turnover. Okay? So basically, mTOR is involved in autophagy, what they call autophagy induction, so the formation of this autophagosome. And then it's important to notice also that once the autophagosome formed, they have to go to the lysosome physically, they have to get with the lysosome for fusion and protein degradation. Okay, so there are two independent steps. One is induction, one is actually autophagy flux, which is the overall protein degradation. So what we did, we measured several of these autophagy-related proteins in the brain, in the transgenic mice, control 15, 18 to 18, and the same pattern flat. And so, well, basically, a long story short, we see an increase in these autophagy markers in both groups of mice. So there is an increase in autophagy induction in both groups of mice. There is a decrease in mTOR signal in both groups of mice. However, only in this case, in these mice, we see less pathology. So again, this dissociation between these two, between autophagy induction and pathology. Here you have to believe me a little bit. So just in so what we're still trying to understand this dissociation. And the hypothesis we're working on it is that, based on data on the literature, is that in AD, in advanced AD, there is a block in autophagy flux. So there is actually an accumulation in autophagosomes. These autophagosomes do form, but then they have some trouble, if you will, and they can't fuse with the lysosome. Okay? And so they just accumulate in the brain of people with Alzheimer's disease and in other transgenic mice. So what we did some confocal work here to see if A-beta can actually get to the lysosome. We done a co-label of A-beta with a lysosomal marker. And so these are the transgenic mice on the control diet and the other two groups of transgenic. So in the transgenic mice with the control diet, there's not much co-localization here where we have to be. There's not much co-localization between A-beta and lysosomal marker. Same thing when we give rapamycin starting at 15 months of age. However, when we give rapamycin at 2 months of age, there is colocalization. So indicating that here we are inducing autophagy in both groups, but only in this group of mice, A beta actually goes to the lysosome. So what we did, we went back and looked at the brain of 15 months old mice. This is the age at which we started the treatment. And we done some EM work, and we actually saw in these mice, this is just qualitative pictures, we have not quantified this yet. But there is a clear accumulation, if you will, of these, into this, what I've been told are autophagosomes. Okay? I'm not an expert, but I just believe what they told me. And this accumulation of these autophagosomes that are hard to find in wild-type mice. So it's consistent with these mice having a deficit in autophagy flux at 15 months of age. So here's the cartoon to explain this working hypothesis. In healthy condition, you have some basal autophagy, basal mTOR activity with autophagy function, and you have this, basically this cartoon is autophagosome that form around amyloid, A beta and tau. And so you have a steady state level. There's not much A beta accumulation. In the early AD, now you have A beta accumulation, and the reason why A beta accumulates is behind this talk. A beta accumulation showed you that it increases mTOR activity. Okay? And more mTOR activity means less autophagy. There's more inhibition, so less autophagosome will form. So there's more A beta hanging around, so it doesn't go, it's not being removed. So there's this, we believe there's this feedback loop. It further contributes. So it's a vicious cycle here between A beta, mTOR, and autophagy. At this stage, if we give rapamycin, now you can induce autophagy, make more autophagosomes, and this will facilitate A beta clearance. Okay. In late stages AD, now there is a blockage in autophagy flux. So you have this autophagosome will form, but they cannot get to the lysosome. So giving rapamycin at this stage, it will increase autophagosome formation, which is it's the data we have. But now this autophagosome cannot be removed, so their content cannot be removed because there's this blockage in autophagy flux. Now again, we are testing this theory. Actually, we have a grant through the VA under review to actually test exactly this theory. So far, I have 5 more minutes. So far, I showed you that basically there's an interrelationship between A beta and mTOR. Higher A beta level lead to mTOR hyperactivity. When mTOR is hyperactive, there's a stronger brake on autophagy, and that feeds back on even more A beta. Show you some of the A beta induced cognitive deficit go to mTOR, because if we block mTOR, you have an improvement in cognitive deficit. I'm still leaving this arrow here because it would be naive to think that all of the A beta induced cognitive deficit go to mTOR. Probably there are also other factors. So what I spent the last 5 minutes is to show you that there is a direct interaction between mTOR and tau. So far, in these transgenic mice we use so far, I showed you when we give rapamycin, you have less A beta, you have less tau. We have showed before that A beta can directly induce tau pathology. So anything we do to these mice, to A beta, if we remove A beta in these mice, we have less tau pathology. Okay? So with rapamycin, we're having less A beta, and therefore we have less tau. So maybe there is no interaction, they would say that there is no interaction between mTOR and tau. Right? It goes to this arrow. Alternatively, there might be an indirect interaction between mTOR and tau. And actually the literature suggests so. And so what we did, we used other animal models to actually test whether there is this interaction between mTOR and tau. One of these is this TSC2 knockout mice. TSC is a tuberous sclerosis complex, and these proteins are a negative regulator of the mTOR complex. So there are actually a clinical mutation in this protein that lead to tuberous sclerosis. This is actually in kids usually they develop these brain tumor, benign brain tumors, and it's related to mTOR hyperactivity. Again, this is a negative regulator. When you have loss of function mutation in this gene, you have mTOR hyperactivity. And people have made animal models These heterozygous TS2 knockout mice have signs of tuberous sclerosis, have learning and memory loss. So what we did, through our collaborator, we got some tissue, brain tissue from these mice. And we confirmed, again, there is more mTOR activity. Again, here I measure PCND6 kinase, the phospholipid of PCND6 kinase. You can see that in the fewer animals we had, there is a significant increase in mTOR signaling or activity. So what we did then, we took the same tissue and measured total tau levels by this tau-5 antibody of phospho-tau levels with this 88 antibody. In both cases, we have an increase in tau levels and an increase in tau phosphorylation in mice that have mTOR hyperactivity. So again, these are yes, in mice, indicating there might be some direct relationship. So what we did then, we bought a transgenic mouse of expressing tau only. So these mice have these neurofibrillary tangles. These are a mouse model of frontotemporal dementia. And again, these have this tau accumulation in the brain. And we set to determine whether reducing mTOR in these mice would clear the pathology. And here's the picture. And when we give rapamycin to these mice, we have fewer neurons that are positive for tau in both hippocampus and cortex. And this correlates with an improvement basically in motor performance. This is the behavioral phenotype of these mice. This is a rotor rod. Basically, we put a mouse in a rod and we measure the latency to fall. It's pretty simple. And the non-transgenic mice in red, with the protocol that we use, they fall about after 20 seconds or so. These are non-transgenic or rapamycin has no effect. Transgenic mice, the tau-only transgenic mice, are significantly impaired in this task. However, when we give rapamycin, we can rescue this behavior. Okay? So I can skip that and go back to my conclusion slide. Again, basically here we show that there is a direct interaction between mTOR and tau. Because if we increase mTOR genetically by removing the a negative regulator, we have more mTOR, we have more tau. If we get tau transgenic mice that are designed to have tau accumulation, and we treat them with rapamycin, so we reduce mTOR activity, we have fewer tau deposits. So we believe that basically this is our working model today, where we believe that A-beta can increase mTOR activity, and this contributes to tau pathology. It also contributes to cognitive decline. I'll finish by acknowledging the people in my lab who've done the work. Mainly Antonella Smith has done most of the work I showed you, and Andrea Magri was visiting a student from University of Catania in Sicily, and he's done some of the tau studies. These are the collaborators in San Antonio and outside San Antonio, and also into the foundations and the NIH for giving us funds for actually doing these studies. And with that, I'll take any questions.
Are you going to use other animal models?
Yes, so I was answering, I think, some question earlier. What we have now, we have mTOR flux mice. And so we are using, rapamycin is an mTOR inhibitor, but it does do other things. And so to confirm that this effect are true mTOR, we're using mTOR flux mice. And right now we bred them with a mouse model of Alzheimer's disease and with a Kim kinase Cre line, so we're removing mTOR just in the forebrain. But eventually, and so those are ongoing experiments. Eventually, we could decide to remove mTOR just in astrocytes or microglia or other sub-regions of the brain.
And so by mTOR, this is kind of outside what you talk about, but mTOR complex 1 or both?
So the way we're doing it's both, because we actually remove the mTOR gene. And so mTOR is part of two major complexes, and so we reduce activity in both.
Right.
And we are, yes,
So
basically.
you can do that later in life, right, with the flux size?
Right. Right now we're doing it, yes, we will do that.
Okay.
Right now we're doing it with this Kim kinase Cre, which is right after birth. Usually the
There
Kim
could be
kinase promoter is.
concerns maybe with neurogenesis because of mTOR2 and development. I mean, maybe, I don't know.
We haven't seen any of that yet,
Any evidence?
but we haven't looked hard.
Yeah.
So as a gross behavioral phenotype, it might
They're developing, okay.
In the autophagosome, you're talking about degrading amyloid, not not A beta.
I'm talking about degrading A beta and not plaques. Plaques would be extracellularly.
Yeah, but it'd be in the form of the amyloid, because you're reducing plaque lesions. So it's been
that's
cross-linked into
So no,
an
notice-
amyloid.
So
Well, what I want to know is, do you know if, when it gets broken down, is it broken down to the amino acid level or to oligomers?
yeah, I don't know. So
So,
let me
so you might have little bags of oligomers
right.
floating
So let me
around,
get back.
which are pro-inflammatory and,
So- There we don't have. So there I know, because the only slide I skipped was we actually I showed you the histology where you have less A beta deposits,
right.
and then I skipped the A beta, the ELISA measurements, where we biochemically measure A beta as monomers. So we have overall we can break down A beta in a tissue extracted monomers and then measured by ELISA, and we have less overall A beta levels. So they could account against having more A beta oligomers. But again, that's all we have done. We haven't looked at them. And so I don't know if the A beta in the autophagosome is oligomeric or fibrillary or just monomeric. That we have, there I have no idea, but we should look.
And what age are you first seeing the cognitive impairment?
In these mice,
Yeah.
6 months.
6 months.
And so when we gave, this is, the first study we did is with 6 months old mice. That's very, it's the onset of cognitive decline. So it's a mild cognitive deficit. There are no plaques yet in the brain, they're just soluble A beta. And when we treat those mice just for 10 weeks, that's sufficient to rescue some cognitive decline. To completely rescue, because there's not much to start with.
So just 10 weeks of
10
treatment?
weeks, yes. And that has been replicated by Veronica Galvan also at the Barshop independently with a different animal model and found the same results.
So there are A beta or precursor protein knockout mice.
APP knockout mice?
Yeah.
Yes, so they are fine. So there are, because APP has what they two homologs, APP-like protein 1, it's called APP-like protein 2. So knock out one of these three independently, mice are fine. If you knock out APP with one of the APP-like protein, either one, APP and APP-like protein 1 and APP or APP-like protein 2, those are lethal.
Is A beta or mTOR in all the tissue or then just the brain? Like in skin?
A beta normally is APP normally is expressed throughout the brain, throughout the body, every cell. In these mice, they express the human mutant A beta, APP, under a neuronal specific form. So it's curious that there are actually there is a muscle disease. It's called IBM, inclusion
body
myositis, right? And where they do, these people accumulate A beta in the muscle, and they get this muscle disorder.
So not in the normal muscle, only in the disease,
Right. So A beta accumulates mainly, I guess, only in this. No, let me take that back. So APP is in every cell. In A beta, there is all the machinery to produce A beta is expressed in every cell. Okay. Then there are people that go to autopsy and they donate their brain after they die, and they have lots of A beta accumulation in their brain, but they cognitively they are normal. And so it's not, this relationship is not clear as it's, people are trying to understand. They come up, each time there's new data, they come up with new hypotheses or trying to modify their own hypothesis.
So is it true about seeing A beta then in other tissues?
As disease goes, the only other disease I know is IBM.
But I mean, do people have the phenotype of IBM without actually having the A beta phenotype without actually having IBM in the muscle? Like
They are,
you see
I'm not
with
sure.
it?
Yes, they are, I'm not sure.
Okay, one last question. Anyone? Okay, thank you very much. I think I got everybody signed in, so just watch your emails for the evaluation. It's a really short evaluation, it's really short.
Reversing Alzheimer's.
You can hear me saying please forever. You're gonna hear me.
Yes,
Okay.
I want to see how entry your improvement. On an annual basis.
I don't, okay, thank you.
I'm gonna chart your improvement over
I'm improving.
10-year graph.
Like the graphics with the mTOR. You keep every year.